Hello everyone. Today we will be discussing on agglutination reaction. It's a type of antigen antibody reaction. If you want to know more detail about antigen antibody reactions, you can go to my previous video where I've talked about it, and I've also discussed precipitation reaction in it. Today we are going to discuss on agglutination reaction. Here, a particulate antigen, that means an antigen which is particulate in nature, will combine with its antibody in presence of electrolytes at optimum temperature and pH, and it will result in a visible clumping. That we will define as agglutination reaction. So in case of agglutination reaction, what is essential feature? That there should be a particulate antigen. So this is the main point on the basis of which we differentiate between agglutination reaction and precipitation reaction. Let us discuss more the difference between the two, which is important to know. First is antigen size. Like in case of agglutination, a comparatively smaller antigen can be detected, while in case of precipitation, larger antigens are detected because they are soluble. So soluble antigens and larger ones. That now when second point is solubility. So I have already discussed that in case of agglutination, it will be a particulate antigen. So it will be insoluble antigen, while in case of precipitation, that will be soluble. When we talk about sensitivity, then agglutination test is more sensitive in nature, while precipitation is less sensitive. So if we want to know the sensitivity of test, and we want to focus on sensitivity test, we will try to find it through agglutination. In case of agglutination, we don't need any kind of media, no gel, nothing is required. While in case of precipitation reaction, sometimes we need semi-solid media or a liquid as a medium. And in case of the basic requirement of agglutination reaction, they are the surface reactions, and so antigen must be exposed. That is why particulate antigen is required, that antigen must be exposed for the antibody, that particulate antigen is to be exposed for the antibody to bind and form visible clumps. What is agglutination? Is just clumping, visible clumps. While in case of precipitation reaction, concentration of antigen and antibody should be equal. That I have explained very nicely in my previous video, in which where I have talked about precipitation. So those who want to know the mechanism precipitation and at how the equal volumes of antigen and antibody are needed for precipitation test, you can go and see that video. So these are the few differences which are important for the exam point of view. I have listed in one slide itself. I try to give notes to my students. And so I would like to know those who are listening to me, whether they are liking these notes or not. And please comment and tell me, you're liking these notes, or you are able to read them. Are they helpful or not? So coming back to agglutination reaction, we need a particulate antigen. That is why it's very sensitive as compared to precipitation. And if antigens are not present in form of particles, if they are not particulate, we try to make them particulate by embedding them on some artificial material like latex or charcoal, and some kinds we use even biological, that is biological material, that is RBC. So we use carriers in this case to make it to make antigen particulate also. And this is the one of the diagram which I have chosen. That is, this is the suspended latex particles coated with antibody. See, these are coated. We have uh, adsorbed antibody on latex particles, so that this latex can make this antibody available to antigen. So if we want to detect an antigen, this latex is helping as a carrier, and we will talk about it later in the other slide also. Coming back to types of agglutination reaction, they are slide agglutination, tube agglutination. Coombs test is also one of the type of agglutination test, heterophile agglutination test, and passive agglutination test. So these are the different types of agglutination tests which I have mentioned on one slide itself. Now, studying these tests individually. What is slide agglutination test? It's the uniform suspension of the sample containing antigen, and it is made in a drop of a saline or a slide. If you have performed blood grouping is one of the type of finding the blood group is one of the type of agglutination test, slide agglutination test. So you have made a uniform suspension of the sample, that is the blood group you want to know, and in a slide with a drop of saline, and then you put an appropriate antiserum to that drop. And if there is clumping, you will say this particular is the type of blood group. That means if you have put a blood group A on the uh, test sample, is you don't know what test sample is, but you have put antigen A to it, then it's like if it's combining antigen A is combining with A, that means antigen antibody reaction is taking place. That means if antigen A is present, then it will combine with antiserum A, that is antibody A. Antigen antibody reaction will take place, and this will define the blood group. So this is a type of slide agglutination test. Then coming to tube agglutination test. In tube, you made different dilutions of the antiserum, and then you keep adding antigen to it. And this is one of the Vidal test is one of the example in which we place 64 tubes at different kind of dilutions, and then we add antigen to it, and we see at what dilution clumping is seen. So this typhoid can be tested by this, and even typhus fever is tested by this method, tube agglutination test. So simple method to find the titer. One more type of antigen antibody test is Coombs test, which was discovered by Coombs, Morant, and Ress, and it was discovered, devised in 1945 for the detection of incomplete anti-RH antibodies. That is the antibodies which are roaming, and they are not clumping at the first sight, like they are present on the RBC, but they are not causing clumping because they are incomplete. And this was, if we add anti-human immunoglobulin to it, these antibodies start clumping. And we will know this is used for screening of when we are transfusing blood, and also in knowing the Rh positive or Rh negative factor present in the blood. This Coombs test is done, it is also called antiglobulin test, because in this we are using human antiglobulin, like antiglobulin against human antibodies. So this is called antiglobulin testing also. It can be done by direct antiglobulin. Coombs test can be studied in two types, that is direct antiglobulin testing or indirect antiglobulin testing. So what happens during direct antiglobulin testing? Let us study that. It's the detection of presence of antibodies attached directly to RBCs. Like this is the RBC in which antibodies are attached, but they are not clumping. But against this RBC, there are certain antibodies which are here present, and these antibodies are causing certain kind of blood related problems. These antibodies, we want to detect them. If we want to detect them, what we add anti-human immunoglobulin, that is antibodies against these antibodies. These are added, and when this is added, if these antibodies are present, then these antibodies will help in clumping, will make a clump, that is agglutination will occur. This happens if we want to detect certain kind of autoimmune diseases. One of the example is autoimmune hemolytic anemia. Other examples also I will be discussing. First, let's clearly understand what is what is direct antiglobulin testing and what is indirect. Now coming to indirect antiglobulin testing, it is done to detect the presence of antibodies present in blood serum of an individual that could attach to red blood cells. That means they are present and that could attach to red blood cells, and that could cause clumping if they are complete. That means this we RH positive father is there and Rh negative mother is there, and mother conceives a baby which is Rh positive, so that Rh positive blood comes in contact with mother's blood, and in that case, in the first baby, what will happen? The Rh antibodies will be formed in the mother's blood. So during second pregnancy, this Rh antibodies can attack the second baby if it's again Rh positive. So there can be a fight between Rh positive antigen that is present in the blood of the second baby, because during the first pregnancy, Rh antibodies, that means Rh positive against Rh positive antigen, Rh antibodies have been formed, and now they can cause problem to the second baby. So this can be avoided by screening if there is Rh positive father and Rh negative mother. So you can do the screening and perform this indirect antiglobulin test. In this, what happens if there are antibodies in the serum, and then this antigen RBC having antigen is added, and then you add anti-human immunoglobulin. This will result in clumping. If these antibodies are present, it will result in clumping, and we can see that certain kind of antigens are present, and antibodies were present in the serum of the individual. So it is a test used for screening blood group, and even some prenatal diseases and prenatal testing can be done. So let's interpret the Coombs test. Interpretation of Coombs test, if it's a positive Coombs test, performed by direct immunoglobulin, anti-immunoglobulin test, then if it's positive, it can be due to autoimmune hemolytic anemia, infectious mononucleosis, chronic lymphocytic leukemia, syphilis, mycoplasma infection, lupus, and it can be a negative reaction to blood transfusion too. So these are the different kind of things that can be present according to symptoms. We can relate them, and these are the interpretation of positive Coombs test. If positive Coombs test is detected during pregnancy, that it indicates that we have to take steps to protect baby from Rh antigen sensitization. That means before the if there is a second pregnancy, precautions have to be taken. Then positive Coombs test in case of newborn indicate there may be anemia, jaundice. So these conditions, Coombs test is very helpful to detect this. Next type of agglutination test is heterophile agglutination test. And what is heterophile? It's like closely related antigens which are common to different organisms. That means certain kinds of antigen may, and what is antigen? Antigen is just chemical combination of different kinds of amino acids and proteins, carbohydrates. Basically, when we come down and we talk about antigen, it's a molecule having different sites, and antigenicity is the unit of where the antibody will combine. So it's a chemical structure. So two different organisms can have a chemical structure which can be similar, or the different sites at which their antigenicity sites where the chemical structure can be similar. And so such kind of antigens are called heterophile antigens, and antibodies that react with these antigens are called heterophile antibodies. This is an antigen, and antibody A, B, C, though we do not classify antibodies as A, B, C, but here A, B, C is written to show that this antigen is having different epitopes where different kind of antigen, different kind of antibody paratope can react. So antibody can be A, B, C. That means otherwise antibodies are of 5 types, and that I have talked in my previous videos. So don't get confused while I'm writing ABC. ABC is just like XYZ antibody, which is present, paratope of antibodies combining with the same antigen. So heterophile antigen is an antigen which can be present in the 2 different species, but they are sharing the same chemical structure. examples of these heterophile antigens are important for exam going students, and that I have put in one slide so that you remember that heterophile antigens can be Frostman antigen, and Frostman antigens are the can be erythrocytes of horse, cat, mouse, sheep, which share the same sites. Some chemical structure is same in them. So, and kidney cells of guinea pig, while bacteria like Streptococcus pneumoniae, Streptococcus dysentery, there are few more bacteria which share the chemical structure, and this chemical structure is shared by all those things which I have mentioned above, and that is Frostman antigens. So Frostman antigens are the antigens we are sharing the same chemical structure, because whatever the antigen is, it is made up of protein. When we come down, it will be made up of some amino acids, carbohydrates, and so, only these things may be present. So some sites chemicals epitopes may be similar in all these, and that is why they are called hetero, means different, but they are same sites are same, so they are called heterophile antigens. So one of the example of heterophile antigens are Frostman antigens, and see so many erythrocytes of horse, cat, mouse, sheep, chicken are sharing the same chemical structure, and same is shared by kidney cells of guinea pig also. So antibodies also get confused, है ना? Then blood group B antigen and E. coli 086 strain share the same some kind of same chemical structure. Blood group A antigen and pneumococcus type 14 capsular polysaccharide shares a similar kind of antigenic structure. Blood group P1 antigen and hydrated cyst fluids share the same structure. Blood group A antigen and streptococcal extract, some of the streptococcal antigenic structures share the same chemical structure. That means some kinds of epitopes are common in them. So these are the some examples for the exam going students to learn. And then heterophile agglutination test is studied during Weil-Felix reaction. That means some protease strains are agglutinated by sera of patients with rickettsial infections. That because the antigenic sites are same, so due to this, we consider that this may be the infection of protease, but same thing will be agglutinated by rickettsial person suffering from rickettsial infection, which is called Weil-Felix reaction. Same is happening during Paul Bernal test, in which sheep erythrocytes are agglutinated by sera of infectious mononucleosis. And streptococcal Mg agglutination test is also one of the example in which patients suffering from mycoplasma pneumonia, that means pneumonia caused because of mycoplasma, and they develop sharing similar kind of sharing with streptococcal Mg group. So these are few examples of heterophile agglutination. Then coming to another type of agglutination test, that is passive agglutination test, in which why it is called passive, because in this we are not having particulate antigen, but we are turning the antigen into particulate, while the antigen is soluble, but we are making it particulate. So that is why this is called passive agglutination test. A precipitation reaction can be converted into agglutination test. That means a soluble antigen is converted into particulate antigen by attaching soluble antigen into with carrier particles such as latex particles, which I have talked in earlier slides also. This conversion is done because agglutination test is more sensitive. If you want to see sensitivity, then and it's more sensitive, so antibodies are detected with more this test is more sensitive, so smaller amount of antibodies will also be. Sensitivity leads to when you have in sample, if there is lesser amount of antibodies or antigen, that can be detected. And even reverse passive agglutination test can be done, and reverse passive is that again you're converting particulate into soluble ones. So that is reverse passive. Now, types of passive agglutination test is latex agglutination test, heme agglutination test, co-agglutination test. Let us talk about them individually. Latex agglutination test, I have already explained this. This is a latex particle, and antibodies are attacked on it, and then they can detect the particulate antigen. So there will be a clumping and cross-linking will be caused. That is latex agglutination test. Coming to heme agglutination test, RBCs, these are the RBCs, and if this RBC is not having a particular antigen, so when we add RBC from outside, particular antigen is not attached, there will be no reaction, and this RBC can be seen in the form of a dot. If there is some virus which is containing heme agglutinin antigen, these enveloped viruses have the antigen which is called heme agglutinin antigen. That means these antigens will combine when come in contact with RBCs, they will agglutinate them. So this, if we want to test this serum is containing this virus, we will put this serum of a patient and the RBCs, and we will see, we will use the test RBCs and we will see the clumping, and then this will be a diffuse, it will not be like this dot, it will be a diffused reaction. So we will see heme agglutination there. While in some cases heme agglutination inhibition is also there, and when this inhibition occurs, it's like if a person is having an antibody against this virus. So when we will, if we feel that this person is already infected with this virus and have, if in the serum antibodies are there, then if we are going to test this, there will be no agglutination, and this is called heme agglutination inhibition. So if we want to test antibodies present in the system against a particular virus, we will see there will be no agglutination. So this is called heme agglutination inhibition when serum is analyzed for a presence of antibodies. Another type of passive agglutination test is co-agglutination. Co-agglutination happens when heat kills Staphylococcus aureus, and that is Co-1 strain of Staphylococcus aureus, Co-1 strain, which has a protein A, is coated with IgG. That means IgG is a type of immunoglobulin present in human. Co-1 strain of Staphylococcus aureus having protein A is coated with a type of immunoglobulin IgG. And then FC portion of IgG. FC portion is crystallizable portion, and if you want to know, it's a fraction which is crystallizable. And if you want to know more about these immunoglobulins and their structures, you can go to my previous videos where I've talked in detail about immunoglobulins. Here, what you remember is that FC portion of IgG gets coated with, combined with protein A, whereas antigen combining FAB portion. FAB portion is the portion where antigen combines with immunoglobulin, and that remains free. So again, what we have done, we have used a Co-1 strain, and for clumping, where I said that for passive agglutination, we have to convert antigen into particulate one. So here we have left the antigen combining portion while we have used a carrier molecule in it. When corresponding antigen comes in contact with this FAB terminal, binds to this antigen, resulting in agglutination. And this is used for detecting various bacterial and hydrated antigen in blood, urine, and CSF. Hydrated antigen is antigen which is having cyst and fluid in it. In case of Echinococcus granulosus, this kind of hydrated cyst is present. In other things also it's present. But here co-agglutination occurs taking the use of Staphylococcus aureus Co-1 strain. So this completes agglutination reaction. This was all about this. Please comment and tell me whether you were able to understand this or not. Thank you.